Hi there, we are Team 13, and for our final project, we decided to focus on osteomyelitis, the bacterial strain. Our group members consist of Miriam Garcia, myself, Andrea Rajo, Kayla Merchant, and Destiny Garcia. For our introduction, we will be focusing on 4 different areas of osteomyelitis and what it consists of. Part 1, History, Prognosis, and Treatment. Osteomyelitis is known to be one of the oldest diseases. It is infectious not only to humans, but animals as well. Osteomyelitis is used to term any bone inflammation or swelling, given that osteon comes from the Greek word meaning bone, myel means spinal cord or bone marrow, and itis means inflammation. The cause of this infection remained unknown for a long time, even though we could identify fossils that presented osteomyelitis, which dated back to the BC period. It wasn't until 1860 that we could identify the microorganisms that cause it. The bacterial organism that causes this is now known as Staphylococcus aureus. There are 3 major categories on how to get this infection. 1 is through the bloodstream. 2 is due to the spread from a contagious focus of infection without vascular insufficiency. This is mainly seen in trauma or surgery due to bacteria gaining access to bone by direct inoculation. And number 3 is contagious infection due to vascular insufficiency, commonly seen in lower extremities due to diabetic foot infection. How does osteomyelitis start? Well, S. aureus possesses on the surface of its cell an arsenal of pathogen-associated molecular patterns that interact with osteoclasts to induce osteoclastogenesis, the production of bone cells that break down tissue and recruit and activate innate and adaptive immune cells. S. aureus kills and takes immune cells during infection by producing non-neutralizing pathogenic antibodies, preventing adaptive immunity to intervene. After recognizing the cells, S. aureus is internalized and escapes from vesicles and persists inside of osteoblasts. S. aureus also has virulent mechanisms, one of those consisting of biofilm formation on implants or necrotic bone tissue and bone marrow. This can lead to bone death, septic arthritis, impaired growth, and giving higher risk of skin cancer. There are a range of different cells and tissues that are involved in this process. All the cells that could be involved consist of neutrophils, T cells, B cells, macrophages, and monocytes. The tissue affected is the bone, the muscles around the infected bone, as well as the bone marrow. The cells found in the bone tissue are osteoblasts, osteocytes, osteoclasts, and bone lining of cells. Osteoblasts are responsible for bone extracellular matrix formation and resorption. Part 2, innate and adaptive immune responses and signaling pathways. What are the first cells of the immune system that encounter S. aureus? Well, we do know that the cells of the innate immune system are the first to respond to bacterial S. aureus. These are phagocytic cells and antigen-presenting cells. 3 of these major cells are neutrophils, dendrites, and macrophages. Neutrophils and macrophages are the major cell type that interact with S. aureus the most, given their abundant presence in the blood. However, this doesn't mean that other APCs and phagocytic cells are not involved. These are just the 2 main cells that focus on destroying and deteriorating S. aureus. So what exactly do phagocytes do? Well, they kill pathogens by various mechanisms, including lysozymes and proteases. They lower the pH of their internal structure to effectively lyse and kill the bacterium. And phagocytosis is incredibly important when trying to destroy S. aureus because this is how we are able to get rid of the bacterium. However, S. aureus has developed mechanisms in order to avoid this. What do the cells of the immune system do when they encounter S. aureus? Well, PRRs on phagocytes will bind to PAMPs on the bacterium. If indirect binding occurs, opsonins need to bind to the pathogen first. And in order to obtain opsonins, we need to make sure that opsonins are able to get processed through the complement system. So mechanisms in the complement system will allow C3 convertase to cleave C3 to C3A, which is a chemoattractant, and C3B to a new convertase, which itself will become a convertase. And C3b is crucial because it opsonizes pathogens once it lands on the surface of the cell. The main PRRs that bind to bacterial S. aureus is the toll-like receptor TLR. So we have three different TLRs. We have TLR1, TLR2, and TLR4. My D88 adaptor protein is the first protein that binds to TLR2 and or TLR1. So my D88 has a prominent role in activating immune responses necessary to control S. aureus. You have an intracellular cascade that leads to AP-1 and NF-kappa beta transcription factor outcomes. Both of these release interferon beta and interferon alpha to activate cytokine and chemokine signaling. So what mechanisms does S. aureus display to avoid detection and prevent its destruction? Well, it has two major types of mechanisms. One of those is opsonization. So extracellularly, S. aureus avoids this by blocking antibody and complement function. The staphylococcal protein A, also known as SPA, captures antibodies via the Fc region, which prevents recognition by the host's Fc receptors. Areolysin, also known as R in S. aureus, cleaves complement factor C3 into a non-functioning C3b. Additionally, our other mechanism is phagocytic, is just a phagocytic mechanism. Intracellularly, S. aureus avoids phagocytosis and escapes by manipulating macroautophagy that happens inside of the cell. S. aureus facilitates the formation of a mesh to protect it from phagocytosis via fibrinogen and fibronectin. It can increase the local pH by producing urease to prevent efficient lysis. Acetyltransferase A, also known as OAT-A, acetylates the peptidoglycan cell wall and allows S. aureus to become resistant to lysozymes. Phenol-soluble modulins, also known as PSMs, enable S. aureus to escape from phagosomes, allowing it to invade the cytoplasm and extracellular space. What signals do they need to send to the rest of the immune system? Well, my D88, IL-1, and IL-6 form a critical signaling cascade. These signal cascades include the recruitment of neutrophils and the containment of S. aureus by producing cytokine signals downstream via my D88 after the TLRs have bound to PAMPs. IL-1 alpha and IL-1 beta will signal on osteoclast cells to increase their viability and resorptive capacities. However, an increased amount of pro-inflammatory cytokines in the area will actually promote indirect osteoclastogenesis. This means that having pro-inflammatory cytokines will contribute to host-mediated bone loss and destruction of bone tissue. Additionally, we have SPA, which is a virulence factor and a surface protein that is located on S. aureus and has a high affinity for IgG immune complexes by binding to the complement binding Fc gamma portion. So blocking the Fc receptor in osteoclast releases NFAT-C1 and various other signals, which does stimulate osteoclastogenesis. What signals need to be sent to the rest of the immune system? It's important to know that B cells secrete antibodies to help neutralize S. aureus and also perform opsonization. However, there's different forms or secreted forms of SPA that can act as B cell superantigens, and they'll bind to their Fab region and induce cell death. A second immunoglobulin binding protein located on S. aureus can also bind to their Fc gamma portion of the IgG on the cell surface, as well as the complement factor C3. Additionally, MHC class II does bind to TCR after the APCs phagocytose S. aureus and lyse the bacteria and then present a bacterial peptide on the surface of the cell. This is important because TCR-mediated activation of naive CD4 positive T cells in the presence of IL-12 does generate Th1 cells. And Th1 cells are important because they do secrete effector cytokines, interferon gamma and TNF, which does further signal the activation of more phagocytes. Additionally, the presence of IL-6 generates Th17 and this also secretes IL-17. What other immune cells become involved and what do they do? Well, as we've already stated, it is important to recognize that we do have both the innate and adaptive immune system working to combat S. aureus. The activation of T cells via APCs is very, very important. We have the CD4 positive T helper cells and their subsets, and we also have B cells. Other cells that are involved are osteoclasts and osteoblasts. Part 3, symptoms and effects of osteomyelitis. What symptoms does bacterial causing osteomyelitis produce in the host? Well, we do have the symptoms, and those include abscess formation, a decreased number of osteocytes, and fever. The signs of osteomyelitis include chronic inflammation, bone degradation, disruption in the immune function, and aches and pain. What effect does the immune response have on osteomyelitis? The immune system mounts a defense to kill S. aureus in the bone tissue, but the effectiveness of the immune system depends on whether the infection is acute or chronic. In acute infections, the immune response can result in complete elimination of the bacteria and restoration of the bone. In chronic infections, the immune response is not capable of completely eliminating the bacteria from the body. Does it get cleared or solved? Well, S. aureus causing osteomyelitis requires treatment to be cleared or resolved in chronic infections. Most treatments send this infection into remission because the infection can reoccur months or years after, and there is potential for reinfection because it is so mutagenic and resistant to specific antibiotics like MRSA. Part 4, important opening questions and the social global economic impacts of osteomyelitis. Is the bacterium S. aureus the cause of infection of osteomyelitis, and does this result in bone loss later? Well, the bacteria strain of Staphylococcus aureus that causes the infection of osteomyelitis is called methicillin-resistant Staphylococcus aureus, also known as MRSA. MRSA can infect and invade the bone due to previous and repetitive injuries, trauma, or surgeries that a person experiences. Inflammation of the bone is what causes the bacterial pathogen to invade that area. This bacterial infection can lead to innate immune responses that trigger bone loss. It is challenging and difficult to treat because osteomyelitis is the bacterial pathogen and it's resistant to any form of antibiotics. There are various forms of treatment for osteomyelitis, such as antibiotics. Penicillin, for example, is an antibiotic that can be used to treat those with osteomyelitis. Any antibiotics must be prolonged for 4 to 6 weeks, though. And certain therapies can be difficult to treat due to bacterial pathogen MRSA and its resistant properties to any antibiotics. However, there's other forms of treatment after the exhausted use of therapies which may result in surgery. ELISA is a laboratory technique that could help us further understand this bacterium. And the way that it does this is its ability to detect and count the number of antibodies, antigens, and proteins and hormones in blood or plasma or even cerebral spinal fluid. And it can also help us confirm certain co-stimulating factors inside of this bacterium. How do individuals who obtain osteomyelitis deal with the social impacts that it causes? Well, we do know that individuals who have S. aureus or osteomyelitis do deal with a lot of pain and severity in their daily life. Additionally, this condition is usually chronic because of MRSA and its ability to become resistant to antibiotics. However, there is an option for surgery, and there are other treatment options and methods that are going to be coming out, hopefully, in the near future. Receiving the proper care and taking care of yourself is incredibly important when you have osteomyelitis due to how fragile the bones become in your body. With that being said, thank you so much for watching, and I hope you enjoyed. Thank you for learning a little bit more about osteomyelitis.